So I probably should also say something about the exam. I haven't really prepared for saying something about the exam. The point is, it will be a written exam. Probably most of you or all of you should know, and the questions in the exam will be very close to what we had in the tutorials, and I'm going to uh, give you a list of the more relevant tutorial exercises next week, so that so some of the things are obviously very maybe not so easy to perform during the exam. For example, constructing a DNA origami structure or something like that will not be a topic, at least not with a computer program. But others are more relevant, obviously, that where you can do a simple calculation or something on a piece of paper. And so I wanted to provide that next week, which is the last week, I guess. So now I should probably start. In the last lecture, we had this part on cell-free gene expression and cell-free synthetic biology, and this is somehow related to the synthetic cell field. In a sense, synthetic cells are also at the core, if you want, of the goals maybe of the Max Planck School Matter to Life, because in a sense, creation of a living cell from scratch would mean that you have achieved the program from matter to life somehow. But it turns out to be very difficult, and also people have tried that already for many decades. And I think that there are waves in of activity where people think that they are now ready to do it, and then they realize it's more difficult than anticipated, and then the next generation has to come to try again. And at the moment, there's still a lot of activity in this field, but I think that there's essentially a diversification of topics and goals in this field, in that sense that some people are looking more for kind of concrete applications of cell-like entities, and usually they give up on typical criteria like being able to grow and divide and evolve. So most of the research in synthetic cells is only on essentially compartmentalized complex biochemical systems, I would say. I hope I'm not offending anybody too much here. But at the same time, this is a very interesting goal, which also helps you to think about like fundamentals of biology and biochemistry, and therefore it's a worthwhile goal, definitely. So one point actually is that in this field you will also find different names for things that look superficially similar. And the point also is that people are not very consistent in the use of these terms. So some people would also have different opinions of what that actually means. And you will find terms like synthetic cells, artificial cells. Many people also like the term proto cells. Minimal cells is something actually different, in my opinion, but that's that would be more like a reduced, originally kind of living cell that would be reduced to its minimal. But there are different opinions about that. So in any case, I think that the proto cell and term, and this is more or less my kind of understanding of it, and you will find other opinions about it. So this is not something that you should try to learn by heart. But I think that most people use the term protocells for cell-like kind of compartments that maybe well, the goal really is more to understand how life could emerge. So it would be kind of a simplified or primitive model of a cell. And often people relate it like in the origin of life field, they or or people interested in origin of life questions would call their systems proto-cells. I think that this is more relaxed in the artificial and synthetic cell field, but here also some people call these things artificial and others call them synthetic cells. And I think that here, in general, people usually talk about synthetic cells when these are kind of compartmentalized biochemical systems that can perform functions that are somehow also performed by living systems. And it should be slightly more complicated, probably, but this is not precisely defined. So in a sense, also in the early days, let's say people just put a cell-free expression system into a lipid membrane vesicle and expressed GFP and called this artificial cell. So at this level, this term is used. So it's not really implying that it can grow, divide, evolve or so. At least not in the usual usage. But of course, with that there are, as I said, different opinions. So there will be hardliners that say these are just cell-like systems or cell-like entities, or they would say you can only call it a cell if it's really growing and dividing and evolving. And I think that there will be different opinions. And the minimal cells is a term that is, I think, more often used when people try to reduce, for example, the genome of an existing cell. So there have been programs where you try to get rid of all the unnecessary parts in a genome to see how far you can go. So this is, but I think that it's also not always completely clear whether it could also apply to a bottom-up approach. There are actually other terms around because I just said bottom-up. I think that some people in the recent years also liked the term bottom-up biology. So rather than sort of building biological systems from the components. I'm also a little unsure in terms of like using either artificial or synthetic. So I asked ChatGPT, which also not necessarily is right, but I think maybe it gives you kind of an average of what people think about it. So I think that the synthetic part obviously has something that's a slightly more like chemical connotation. So at least ChatGPT says something like the term synthetic typically refers to something that is made by chemical synthesis, especially to imitate a natural product, blah, blah, blah. Artificial generally refers to something made by human beings rather than occurring naturally.
I think it's funny that he used the term artificial to describe synthetic.
Yeah, well, that's the problem. So in a sense, of course, if something is man-made, how would you make it rather than using chemistry? I don't, it's kind of, okay, whatever. We will spend the rest of the lecture if we start this. Okay, but in any case, it's, I would buy that artificial has a broader meaning. And I really think that I know that many colleagues prefer the term synthetic cells. This is why I also use that as the title, because it somehow emphasizes the role of the person that builds the system using synthetic methods. But we will see. Well, I will come back to that a little later. Um, so one question is, of course, why would one like to do this in the first place? So, and as I already said, there are different people in the field with different opinions about that. So some would like to simply understand, uh, maybe come up with a realistic model for the first potential compartmentalized self-replicators. And others would be interested in understanding kind of how the transition from non-living to living takes place. And this could actually be the same, but it could also be different in that sense that the origin of life people have to somehow think about what would have been the conditions on early Earth that could give rise to such a compartment. So you cannot use certain components that are known, not that where you know that they have not been there on early Earth. So this is slightly more restrictive. You need to build your protocell with, let's say, if you want to have a membrane, you can only use molecules for the membrane that have been that were already there. But if you only want to create something that is living, then apart from the problem that we don't have a good definition of life, the point is that here you could also use other components. So you could, let's say, you could make a membrane from synthesized polymers, that like a polymersome. And if somehow the system that you create fulfills the criteria of a living system, like growing, dividing, evolving, maybe, then you could say that this is alive, but it's not a protocell in the sense of this could also have been realized on early Earth. So that's kind of the difference, I guess. But of course, they are related. In terms of the definition of life, I don't want to I don't give the definition here. So many people resort to the NASA definition, and that was essentially, I think, something what I just said, like a chemical system capable of growth, division, and evolution. And another definition, for example, I read Kate Adamala, who's kind of well known. She originally was in the Shostak lab and now works on these problems. She said, she says, for example, I know when I know it when I see it. So that's also a very good definition. I think that's the definition that most people would probably. Okay. Um, then, of course, this is the fundamental thing. And the but there are many people who really just see these systems as interesting chemical systems that are just maybe very powerful or more powerful than non-compartmentalized biochemical systems. So they could be like sensors, advanced sensors interacting maybe with living cells or production like machines. Soft robots is sometimes also mentioned in this context. So that would be the more practical approach, where I guess in the end it comes down to the point that certain biochemical functions require compartmentalization and spatial organization, or to improve the functions, it is useful to have compartmentalization. And then in the end, what you create simply out of necessity somehow looks cell-like, because this is the best way to do it, maybe. So okay, we will see. So the rest of the lecture probably more contains like examples of these different things, and it's difficult to. I don't really see a extremely like a very clear, let's say, theoretical like approach here. So people just try out things, and of course one has certain ideas how to make these things, but it's relatively diverse how people approach the problem. So I just give a few examples, but there is a lot of papers out there and which all kind of look slightly similar. But one thing, of course, I guess, is that in the end, most of the things are trying to mimic natural biochemistry. It's not that anybody, or at least I'm not aware of, radically different approaches where the system would look very different from what we already know from existing living cells. And probably it's difficult to think about that, how life could look like completely differently from what we know from biochemistry. So one famous kind of abstracted scheme for what such a protocell would require is the famous chemoton by Ganti. And if you look at this essentially is, as I said, an abstracted model of what you find in cellular biochemistry anyway, because there are several interlinked reaction kind of cycles that are used, or that you need to make a growing and dividing compartment that contains some type of information bearing polymer. And in the end, of course, you can directly see that this is our like DNA replication, some metabolism here, and that's the membrane, and it's somehow linked. So the idea is the following, that for example, here is a template replication process that's like DNA replication, where there is a template, the polymer with some monomer V, and the V, so this PVN templates the growth of another polymer that has the same length, and then it splits in two. So that's DNA replication more or less. But then also this PVN somehow informs the production of other macromolecules, and this is shown here. There is, for example, this R here, there's the V that goes into this cycle, and that is supposed to be some type of metabolism. And you can see that because there are kind of some kind of molecules called XA, they come in over the membrane, and then they are used here to make stuff. This is this metabolic cycle here. And then some of in this metabolic cycle, some of the products are actually used to make more of the T, and the T is the membrane component. And the Y is probably a waste product. The cell needs to have a kind of a outlet for the waste, because you need to operate the thing in out of equilibrium, which means that you have like high energy compounds coming in here, low energy compounds going out. So the whole system, so this cycle here is driven by the, in a sense, non-equilibrium state. And it's coupled to replication of the genetic template and the growth of the membrane. So this is the idea. But of course, this doesn't tell you how to do it. So in the end, the task would be, if you say, I don't want to use biological component, you need to come up with a chemical system that does the same thing, and which is challenging. But of course, people try it. Oh, here I have sound. I need to switch off the sound. Oh, I don't know how to switch off the sound, so I'm sorry. Okay. But this is just a computer animation. It doesn't really tell you how to do it. It's more like the imagination of the people who made this video. So this is how the thing should in the end look like. And that's more or less like an implementation of kind of this chemoton. This animation shows the process of protocell growth and division. RNA nucleotides there must be some process that leads to the growth of the cell. And in this case, you actually see these little micelles coming in from the outside. that would lead to membrane growth. So with this, they circumvent the necessity of producing the membrane within the cell. And then into daughter protocells. With the contents of the original protocell, you need some mechanism, obviously, to copy the genetic template here, and then there must be a mechanism to separate the two, the bigger compartment into two, and there must be a mechanism that actually ensures that you have like one copy of the genetic template in both of these. And it's not clear how that would actually work. So this is therefore a scheme, of course, that how people imagine that. This is already like a relatively old paper on that, where by Jack Shostak, where they thought a lot about how to maybe implement like just the basic growth and division of the compartment. And this is kind of this growth by external feeding. So the idea is that somehow you would feed the membrane monomers to this compartment. It would grow, and then some mechanism would lead to division. And here they suggest shear forces or thermodynamic instability. So both of these things actually do occur. So there's so also membrane biophysics has a very long tradition in also studying kind of maybe some shape transitions in membranes, and it could be that under certain conditions there is a instability that makes a big kind of vesicle energetically less favorable than having two small vesicles. But also you might be able to sort of induce the division by shear forces, and therefore people have thought about whether this could occur, let's say, in porous systems where there is a strong like flow of medium that sort of will rip apart little components. Here is an old paper from Garching from the times when Erich Sackmann was the only biophysics guy here around with Joseph Kies on this paper already in 1991, where they actually studied such division processes. And in this case, they actually used an osmotic shock to somehow induce cell division. So you see that this can be done with like model phospholipid membranes. But then in the end, the really big challenge is how to somehow couple this to such a process as it is shown in this video. So you can, and maybe that's a general problem of the whole field, that you can show many kind of aspects of the overall scheme. So you can show replication of DNA, or division of a vesicle, or all these aspects. But it's difficult to couple all these things, so they sort of synchronously kind of occur how you would like that in these coupled kind of chemical cycles of the chemoton. So here is another paper by Jack Shostak, where they in the past they actually used fatty acids, and partly with the argument that maybe phospholipids were not present on early Earth. So, but there are kind of arguments why fatty acids would have been there. And what they studied here is they actually looked at clay minerals, and what one can actually see is that and they specifically used this montmorillonite. So the argument is that there is a lot of such clays or such silicates on Earth. What they found is that apparently when you have a film of fatty acids on the clay and you wet that, then you form actually like vesicles. And if you also have RNA present by chance, then you will also generate like little fatty acid compartments that contain the RNA. So that was just like one finding where they would want to say that in principle, compartmentalized systems with RNA inside could emerge in such a setting. So one of the reasons why they actually used fatty acids also has to do with one of these many chicken and egg problems that, for example, modern phospholipid membranes are rather impermeable. So you need usually membrane pores, for example, to import more complicated molecules into phospholipid membranes. In particular, it's difficult to get any charged molecule across the phospholipid membrane. And but the problem is that when you need membrane proteins, you already need transcription translation in place to produce the proteins. And therefore, the argument by Shostak and co-workers is that you need a more permeable membrane in the beginning. So you have compartmentalization that still allows you to capture macromolecules, but allows exchange of small molecules with the environment without any proteins present already. That's a difficult problem, but that is kind of the point here. So one idea is, and so for example, if you, and that's another point, of course, you need something that self-replicates, and many people have studied different self-replicating systems, but like for example, one of the most famous systems of is the Q-beta replicase. You probably heard about that. That has been studied by many people, where you have a replicase, and which is a protein that replicates a specific type of RNA molecule. And with that you can study kind of replication of an RNA molecule, but it obviously requires this protein here. So people were looking for RNA-based replicators where RNA can replicate itself, and that is challenging. But apart from that, if you had something like this and you would compartmentalize that, then you can see under certain conditions that, for example, this polymerization of an RNA molecule changes the osmotic pressure in such a system, and that leads to vesicle growth. So the idea is that, for example, if something replicates faster, or the replicator or the replicase is faster, the compartment will grow faster, and then you will have cell division in a sense by some mechanism, and you will in the end have more copies of the faster growing ones, and that already looks a little like Darwinian evolution, because in a sense the fitter protocells are the ones that replicate faster, they grow faster, divide faster, and then they somehow compete for resources with the other slower ones. And I guess that this is generally the hope that in this field, that if somebody comes up with a replicator and compartmentalization and couples these somehow, then evolution will do the rest. So you just need to kind of barely achieve this basic mechanism, but then evolution kicks in and will make your system better and better all the time. Yeah.
But I mean,
it happened once on this.
But I mean that we have to wait our 2 billion years. So,
Yes.
I mean that's unachievable for the land.
Yeah. Okay, maybe, of course, today with lab automation and artificial intelligence, we will make it in only 1,000 years, maybe. Okay, I have no idea. But that's certainly a challenge. I guess that Okay, I'm not an expert here, but I guess that of course the conditions might have been okay. That's also a big question in the field. Were the conditions very special and it was a rare event, or is life something very generic that will always occur because it's a robust kind of phenomenon that will. So it's not clear. I don't know, and and I could imagine that with technical support you can do it faster, but it's not that somebody has achieved it, so I cannot really say anything about it. But I have to say that we actually have something like this here at the TUM. I'm also part of something called an innovation network, and we call it RISE, Robotic Intelligence in the Synthesis of Life. That sounds cool, but we have not achieved it yet. And this is led by Job Böckhofen. And I think that the idea was to really speed up such processes. The problem is that you do not even know where to start because we simply do not have the replicating system or in place to perform these things. So here is one example where it at least one part sort of worked, and that's a Nature paper by Sheriff Manzi with Jack Shostak, where they actually had such fatty acid membranes. So these are the more permeable membranes, as I already mentioned, and you see monomers uh, on the outside. This includes the fatty acids and the nucleotides. And here's the important point: the nucleotides would not enter a phospholipid vesicle. So that only works when you have a fatty acid vesicle. So it somehow squeezes through the membrane here, and then you can, in this case, they have copying of a template-directed copying of DNA inside of the cell. The cell apparently grows by a change in osmotic pressure and divides, and they can show that there is some primer extension, and this is what is a typical experiment in this field, that you start with a primer, and then you see that there is nucleotide by nucleotide extension in the gel over some time. So in this case, what these people do is you usually try non-enzymatic template copying, because at the origin of life you had no DNA polymerase. So you add chemically activated nucleotides to sort of do the polymerization, and this is extremely slow. So even if you see that the time scale, so after 24 hours you have like some decent copying of a relatively short piece of DNA. And that's a problem, of course. And there are many problems related to this, which I don't have time to talk about that. But for example, let's say there will be, let's say at the origin of life, potentially there was not DNA but RNA, and RNA is chemically not extremely stable. So if for example, the time scale that you need for polymerization is so slow, then the RNA will be degraded before it is produced, or while it is produced. So it's difficult to really come up with systems that work. And this is an ongoing research. It's a very interesting problem, of course, and very challenging. And the problem is that in the end, the whole field, of course, can only give plausible scenarios for the origin of life, and it's difficult to imagine how you could prove that something has happened. But you can say that that the conditions were there or that it was very likely to happen. Yeah, here the membrane thing I already mentioned with the problems. So one interesting other aspect is that ATP synthase, of course, is a membrane-bound protein, and you need ATP synthase for making ATP. So ATP synthesis you find it in all life forms. It's a very ancient protein, and so for this you need a phospholipid, and it also is difficult to imagine how to, well, modern biochemistry somehow requires the production of ATP. And of course, what usually the argument would be, maybe there was like a different type of self-replicating system before, or a different um, uh, type of biochemistry, like a few billion years ago. And then there was an evolutionary invention, and that was so successful that it just took over. And we do not have any fossil evidence for the previous life forms. And therefore, it's just not, it's very difficult. It's a very difficult problem. So here is actually a paper from a few years ago where Jack Szostak now had the idea to maybe somehow use mixed lipids. And the idea was that you could maybe start with fatty acids and invent phospholipids, and then somehow the membrane sort of would be replaced by a phospholipid membrane during evolution. And here, this is a scheme of the co-evolution of the enzymatic processes and phospholipids. And so what you see here, so this would be the phospholipid, the fatty acid, and that would be the phospholipid. And what they assume is that there might have been a ribozyme that catalyzes the conversion of a fatty acid into a phospholipid. And what they also claim is that the phospholipids, then if they provide some type of evolutionary advantage, maybe there could have been this sort of co-evolution, that the compartments that have a better ribozyme, that make more of the phospholipids, would help the biochemistry in the compartment. It might protect it more, and therefore better ribozymes would sort of evolve, and therefore more phospholipid membranes and so on. So that somehow I think is the idea to kickstart sort of the production of phospholipid membranes starting from a fatty acid membrane. What you also see here is that in this graph, they show that the phospholipids actually stabilize the fatty acid vesicles. So they put in DNA into the vesicle, and they measure DNA oligo retention, so how much DNA is retained. And you see that these fatty acid vesicles are actually quite sensitive to magnesium. So if you add some magnesium, then the oligos just leak out. But now they add a little of the phospholipid, and at above a certain percentage of the phospholipid is POPC, you see that also at higher magnesium concentrations, this is more stable. Now, magnesium is important because you usually need it for polymerization of DNA or RNA. And with this, they sort of achieved this coupling that you need a slightly more stable membrane in order for the sort of the copying to work. But if you have an unstable membrane, then you cannot supply the magnesium, and therefore there's kind of this coupling between making phospholipids that stabilize the membrane. You can add more magnesium, the replication becomes more efficient, and so on. Yeah, it's a complicated topic. So therefore, you can forget all of these things when you deal with artificial and synthetic cells, because then it's not, then you can do whatever you want. You don't need to use fatty acids, and you can use modern biochemistry. You are allowed to use polymerases and so on. And then things become maybe slightly easier. And there have been many proposals how such a cell should look like. And here you see that the term minimal cell is used in order to increase the confusion. And what they sort of suggested that a minimal cell should look like this here. And now you see that they use modern components, for example, transcription translation here to go from DNA to RNA and then to proteins. Then they have ATP generation. The idea is that this would be some intracellular vesicle that has bacterial rhodopsin. So you shine light on that bacterial rhodopsin that generates a pH gradient across the membrane, which drives the production of ATP with the ATPase. And you have some membrane channels here. So this is a suggestion how like a sort of simple cell with metabolism could look like. And here you obviously would need proteins and things that are already kind of out there. And similar things have actually been achieved in the meantime, of course, since this proposal in 2002. Another kind of related question is how many components do you actually need for the minimal cell? So whereas this paper makes just such a sketch, other people try to write down lists of which components you actually need. And here the idea would be that maybe you could actually make all these components, purify them, put them together, and then hope for the best. And in this paper, and you see that this contains many things of the chemoton again, but here they actually do not consider production of the membrane. So they assume that there is already a membrane that lets small molecules across the membrane, and they mainly look at the internal networks here, which is essentially the central dogma again. So here you have DNA replication, then production of RNA, which means transcription, and then the kind of translation takes place down here. So here several RNA molecules are made. So the mRNA, then the RNase P. RNase P is something that you need for the production of tRNAs, for trimming RNA. That's also an RNA molecule. And then the yeah. And I actually have difficulties to see in such a scheme whether sort of this is, so yes, this will work or it will not work. I don't know, there are many arrows, but what they actually did is they made a list of what they think they would need to set up such a system. And you will, and now that kind of is related to what we talked about in the last lecture, that this more or less, that is very similar to the contents of a cell-free expression system. And so they say we need a DNA polymerase, and I'm not sure what the recombinase is needed for. That's here. That's maybe for making rearrangements in the DNA. I'm not totally sure about that. Then they choose T7 RNA polymerase for the RNA production. then RNase P, as I said, is required somehow for the making the tRNAs. Then the ribosome, and the ribosome contains the ribosomal subunits, the proteins and the ribosomal RNA. And then the translation, initiation, elongation, and release factors that we talked about, and the tRNAs. And they made some considerations here. For example, you don't need all the tRNAs that are possible in principle, so you can work with a reduced set probably, and that has to do with the also with the redundancy of the genetic code, that not all of them are actually needed. And they also have they considered chaperones here for assisting protein folding. And in their calculation, they come up with the requirement for 151 genes, and which would code for 38 RNAs and 113 proteins. And so as George Church is on that paper, of course, I think that from his perspective, this just means that this is a piece of DNA that you could in principle synthesize. So one could in principle make the genome for this system by synthesizing the DNA. And then you would somehow need to start the whole process. And then so that's certainly an interesting approach, that you think about what you really need as the minimal number of components, and then maybe you just make the DNA, put it with some cell-free expression system to start it into a compartment, and then see what happens. One has to say, however, that nobody did that so far. So there must be technical issues. Yeah, I think I'm going to skip this. So in terms of compartmentalization, I already said that there are trade-offs between different types of compartmentalization. And maybe I should also say that not everybody says that you actually need compartmentalization for a living system. So one also needs to somehow separate the question of transition from matter to life, or maybe the origin of life and compartmentalization. So some people in the origin of life field believe that there were already kind of self-replicating, maybe chemical systems that were not necessarily compartmentalized in a membrane. And this might have, so maybe there was like a self-replicating system in a typical scenario would be in a little, like in porous rock somehow. And so the pores of the rock were the compartment, and then maybe later this system was encapsulated by some type of membrane. So therefore one also needs to somehow differentiate that a little. But if we talk about cells, I think that usually people think that this should be a compartment, and then here we have different options. Many people actually like to work with emulsion droplets, and these are of course not like encapsulated, not systems encapsulated by a membrane, but it's because it's usually something like a water in oil emulsion with a surfactant. And therefore, in these cases, you do not really have exchange of materials with the outside world. But on the other hand, these can be very stable, and you can make a lot of them. And of course, droplet microfluidics is something different than creation of artificial cells, but you can test many ideas that relate to, for example, compartmentalization of chemical systems also in emulsion droplets. And if that is sufficient for your application, they are maybe the best system because they're really stable. You can make many of them and so on. Then, of course, phospholipid bilayer vesicles would allow you to incorporate membrane channels, and they obviously are more cell-like, but they are usually more difficult to prepare, not so monodisperse, which might not be a real criterion, and less stable. You also need to see that, of course, modern cells are not just lipid membrane encapsulated. There is a cell cortex, or bacteria have additional kind of cell wall. And so if it's about the stability, then one idea certainly is to also create like a more stable kind of compartment. And this is also what people have already done. So it's not that this hasn't been done, but these are the things you need to consider. If you're a more chemically minded person, you say, I don't really care about the similarity to biological systems. And there's also polymersomes, so many like polymers, amphiphilic polymers are known to make nice membranes, and they can be actually more stable than phospholipids. You can also do the same thing with peptides. There's also something called proteinosomes. So you do not necessarily need to use lipids. And in the last years, maybe 10 years, there has been a lot of activity on membrane-less compartmentalization. And I'm not sure whether I'm going to talk about this in the last lecture next time. I'm not decided yet, but this is a kind of a very hot topic in cell biology anyway, but also one of the models for compartmentalization, also in relation to the origin of life, was based on membrane-less coercivation. And so the advantage of something like this, and essentially this is something like phase separation. So you have a mixture that demixes, and you have two different phases. And in one phase, typically certain macromolecules are concentrated up, and they are spatially then sort of separated from the other components. But because there's no membrane, you can exchange things with the environment without any problem. So that is an advantage of them. Disadvantage is that you cannot do something like ATP generation with ATPase because the ATPase needs the membrane. So therefore, yeah, also difficult. And now I'm going to just show you a few examples of what people have made in this field and these are more or less randomly chosen examples, but some of them are actually from researchers in the Matter to Life network. And here's like a big paper from 2017 that has many of the people on it, like of course Joachim Spatz and Kai Sumbacher, Reinhard Lipowski, Ebert Bodenschatzer. So this is like a list of PIs from Matter to Life. Jean-Christophe Barré is a well-known microfluidics expert, and they actually came up with a method to make big vesicles in a microfluidic system, which is actually also challenging. So here, as I said, the problem with emulsion droplets is that you have water in oil emulsion and a surfactant, and that's not a lipid membrane. And what they did here is they made like this water in oil emulsion, and these molecules here, that's a typical surfactant that contains PEG or polyethylene glycol as a hydrophilic head group, and a fluorinated polymer as an omniphobic sort of tail, and that makes very stable and very nice emulsions. And what they actually could do here, they actually attach gold gold nanoparticles at the inner surface, and they also encapsulated small vesicles. LUV stands for large unilamella. So these are on the order of 100 nanometer diameter, maybe a little bigger. Well, here's a scale by 100 nanometer, and these can actually grow to become bigger vesicles, and then they are called GUVs, giant unilamellar vesicles. You see them here. And addition of magnesium then leads to actually destroys these GUVs, so they are not very stable with respect to magnesium, but because they are compartmentalized here, somehow that outer construct here sort of templates the membrane, so they form like a giant unilamellar vesicle that is actually stabilized by the droplet. And all of this is done actually in this microfluidic system. So you first you generate these droplets, and at one point they can actually break the emulsion, and breaking the emulsion means that you somehow strip off the outer part, and then you actually get vesicles that are nicely formed. And what they could achieve here is, for example, also create something like a cytoskeleton. Here are images of microtubules, an actin cytoskeleton that's and they use the technique that's called pico injection. That's actually a nice technique. Unfortunately, it's not shown here. So you can actually, with an additional little channel, you can sort of inject something into an emulsion droplet with a short electrical pulse. You can sort of fuse a droplet with another droplet coming in, and then sort of you can add additional components. And one interesting thing here is that they actually generated ATP inside of these systems. And for this, they actually acidified the oil and put the ATPase, the FATPase, into this membrane. So these little like red, blue dots here, they are ATPases. And because the oil is sort of more acidic than the interior, you have a flux of protons going actually through these ATPases and generating ATP. So you see that already kind of quite a number of components are in place in this system. You have ATPase, you have a cytoskeleton, you have a membrane. In this case, there is no genetic template inside, so it's really constructing more the kind of the protein and metabolic components. And maybe this is sufficient for certain applications. So in a sense, you don't, if you don't want to produce more proteins, you don't want growth and division, and maybe more use this as a functional container, then that's also a good approach. Here is another paper by Johannes Spatz, actually with Kerstin Göpfrich here, already also some years ago. But what I find nice in this specific example is that they also incorporated mitochondria. So that could also be a trick in a sense, that you actually incorporate like real bacteria or real organelles as like organelles inside of a synthetic cell. So here is E. coli, mitochondria. So they could provide some biochemical functionality, but then obviously this is not completely bottom up anymore. So you take kind of the really complicated things you just sort of still take from biological systems. So here is a paper by Petra Schwille. So she's usually not dealing with genetic processes, but she is very interested in cell division, in particular bacterial cell division. And she has been working on that for many years. And there is one system that she characterized extremely well that involves these bacterial cell division proteins min-D and min-E. And I cannot explain the details here, but the min-D-E system is a system that contains a membrane-based component, and one that is in solution, and there is a phosphorylation cycle of going on that leads to actually wave-like sort of dynamics on the membrane in the case of bacteria. And for the bacteria, this plays the role that this is like an oscillation going back and forth in the cell. And that helps the cell to actually decide where the sort of the center of the cell is, where to assemble the fission ring for the division of the bacteria. And this is kind of vaguely similar to a real oscillator. So if you think of like a pendulum, then the pendulum spends most of the time at the at the positions where it's most elongated, and the highest speed is when it goes through the minimum. And if you sort of convert that, then of course that's only a rough picture, but if you convert it into concentrations, then the concentration of your pendulum is higher at the kind of when it's completely elongated, and the concentration is lowest in the middle. And if you would couple such a concentration to some other chemical process, then that would, for example, allow you to position something exactly in the middle or exactly at the pose of the cell. So I think that's kind of the idea behind it. And the interesting thing is that this also works outside of the cell, and it works in vesicles. So if you inject only these two proteins here into such vesicle systems, and that's another method how to make vesicles. So very popular method to spin using a centrifuge water in oil kind of droplets through an interface. So you first actually generate such micelles or inverted micelles that are covered by a monolayer of lipids, and then you spin them through the interface that contains additional lipids, and the compartments on the other side in the aqueous part here, they are sort of coated by a lipid bilayer. And they also encapsulate the macromolecular components. That's actually another big problem in how to encapsulate complex biochemical stuff in such vesicles. I haven't mentioned that before. So one of the reasons why they make this rather complicated approach here is that giant vesicles are rather fragile. So if you have a giant lipid vesicle, you cannot easily get something inside. And we already saw that there is also some sensitivity to buffer conditions, magnesium, and so on. And this is sometimes not compatible with these vesicles. And therefore, approaches like this here allow you to encapsulate stuff during the production already. And here they see that these vesicles actually start to kind of oscillate. They make these dumbbell forms, and then here they actually divide and then fuse again, divide and fuse. That's quite funny, and at least it shows you that somehow maybe cell division could actually be achieved in such systems. So here's a rather complicated system that is very interesting, that actually uses, in a sense, components from a photosynthetic system and ATP synthase to generate ATP inside of such a compartment when you shine light on that. And it might, I need to slowly explain what's going on here. So there is actually two types of vesicles. So one big vesicle, that's here, the giant vesicle shown in red, and there are small vesicles, these green guys, and these actually look like this here. And the idea is that you have ATP synthase in these small vesicles, and there is two different light sensitive channels inside of the inside of the membrane. So there's proteorhodopsin, and proteorhodopsin actually pumps protons across the membrane, and this depends on shining light on them. So if you shine green light, then the proteorhodopsin will actually allow the protons to pass. If you shine red light, it's inactive. And they also have photosystem 2 here, and here the action is actually the difference. So when you shine light on red light on the photosystem 2, it will actually generate protons. And in this case, the protons will actually go through the ATP synthase and make ATP. Whereas when you shine green light, then there's no production of protons, and you drain all the protons through the proteorhodopsin. So therefore, there's some kind of light control that you can generate ATP by shining red light, and you stop ATP generation by shining green light. That's the idea behind it. And now they actually add something that binds into the membrane that also allows ions to go across the giant membrane here. And they use the ATP to polymerize actin. And the actin actually makes something like a cell cortex. So it actually deforms the lipid membrane, and the overall system that looks like this. And I think that's actually pretty impressive. So they have this kind of big membrane here, and they actually allow when they shine light on their system, they actually see this growth of such a actin kind of network inside of these systems. And yeah, very different topics. So you see that, like, and this is what I meant in the beginning of the lecture, that it's quite diverse, that people do very different things. There's a question.
Sorry, but why are we interested to control this like acting growth with light? So
Because growth is cool, and control is cool. No, I think that, of course, I don't know, maybe I guess that this is related to cell motility. So if a cell moves then on a substrate, then it also reorganizes the cytoskeleton all the time. So in principle, I guess you could think of something where you shine light on one side of the cell, so it's polymerizing actin at one side, so it's polarizing the system, and maybe it crawls in one direction. I think that's so it's more like it's a robotic application, maybe. The other thing is, well, I don't know, it's sort of, I think, combining many things that are somehow interesting, in that sense that there is some metabolism with the ATPase, and there's production of of this polymer, and there's a mechanical So it's some type of coupling of different processes. I think that's the so in a sense it's obviously more than making GFP inside of a vesicle. So that's I think that's the main interest behind it. Here is another paper, and that is a little more related to. So you see that the previous examples didn't have any DNA replication, but you will need DNA replication for making a self-replicating cell that could also evolve somehow, and that's also a very challenging problem. And here they actually used a genome from Phi-29, and Phi-29 actually has such a kind of a linear double-stranded genome, so it's not circular, and there is a replicase that actually can actually start replicating the DNA from one side here, and that's indicated here. So that is one system that is sort of interesting, and that you could potentially implement a self-replicating genome based on that mechanism. So what you would need in principle is to also encode that DNA polymerase onto the genome itself. And this is what they tried here, that they encoded different kind of components that you need for that on such a construct. One of them, for example, is single-strand binding protein here that sort of prevents rehybridization and other things. And here they actually achieved or tried to achieve this DNA replication inside of a liposome. So that's just the one example, but there are many others that actually work on specifically this problem to get a self-replication cycle for DNA or RNA running in such systems. And this is actually based on the pure system. So therefore, this is not it's not a proto-cell project. It's not an origin of life project. It's really a project that tries to make a self-replicating system using kind of modern components. So here is one. I'm just putting this up because it's somebody from the Matter to Life network, Katharina Landfester, and co-workers wrote actually a review about that, and from 2022. So there's a lot of kind of different stuff that is sort of related to this compartmentalization problem, and it's getting a little difficult to have the overview. It's in a sense, it's similar to DNA origami research. So DNA origami has also been used for all kinds of stuff in the meantime, and therefore it's somehow not possible really to talk about like all of the different types of variations and applications that are out there. So if you're interested, maybe you want to look at something like this here, and it also somehow discusses many of the different aspects why people are actually doing it, and the approaches that are taken here. And one thing that is maybe interesting to note is that this review is called Synthetic Cells from Simple Bio-Inspired Models to Sophisticated Integrated Systems, but it's in the section Artificial Cells. So you see that there's like big confusion out there. And the other thing that we also just talked about, it's if you're not really interested in origin of life or transition from matter to life, you can also look at all of these systems just as robotic systems. And here is one paper that I found that just came out like in this year, 2024, and it's called lipid vesicle-based molecular robots. But if you look at this, of course, this is exactly the same like the synthetic cells that we talked about. So it's more like a matter of your goals or your background, maybe how you see that. And I think that I would actually also support this type of view, but because what you are interested in the end is a chemical system that somehow interacts with its environment. It can do stuff, maybe perform some simple calculation, produce molecules, maybe move around. That's a robot in the end. And I think that's more if you interpret these systems as little robots, what the advantage of that is that you are not really sort of so confronted with this question, is it alive or not, or do I need to implement replication or not? It's more like a complex technical system built with biomolecules. One paper that had some prominence maybe in the last years is this paper, and by Stephen Mann. Stephen Mann worked a lot also on coazer weights and similar systems. And here the paper is called Living Material Assembly of Bacteriogenic Protocells. So here he actually prefers to use the term protocell, but on the other hand, this is really not related to the origin of life. So you see that it remains difficult. And what he did in the end, I think, is that this is more like producing a cell extract in situ. So rather than using the pure system of making like a bacterial cell extract, in this case, what they actually did is they captured bacteria in a actually droplet, and then they lysed the cells so that only the interior of the sort of the biochemical machinery of the cell remained. And so they used coacervate droplets, and as I said, phase separation is a hot topic, and one mixture that actually produces little droplets is this mixture here of this polydiallyl methyl ammonium chloride and ATP. So so to my knowledge, so this here is a positively charged polymer. The chloride is just a counterion, and this positively charged polymer somehow interacts with the very negatively charged ATP, and that makes like a very viscous kind of droplet-like stuff, and that is indicated here. And it turns out that certain things are simply absorbed by such a material. And in this case, they used E. coli and Pseudomonas, and the Pseudomonas is abbreviated PAO1 here. And somehow the E. coli ends up inside of these vesicles, inside of these droplets, and the Pseudomonas somehow ends up at the surroundings. So what they did here is doing all kinds of making impressive kind of images of the process. So here they actually incorporated the E. coli into these droplets, and that's kind of a cross section of it. You see the green E. coli in this droplet, and here they actually absorbed the Pseudomonas, and apparently Pseudomonas only sticks at the outside and doesn't go inside. I'm actually not sure why that is the case. And here they then actually used bacteria like Pseudomonas expressing M. cherry and bacteria expressing GFP, and assembled both of them on that droplet. And here's how the cross section looks like. So in a sense, you have many E. coli in the interior, and it's like a membrane made from pseudomonas. And as I already said, now what they did is they actually lysed the cells inside of in this droplet. And it turns out that the well, that they have some lysis protocol here using lysozyme and melittin. And what you can see in these images is that here are the bacteria, they are still intact. So that's and then they actually lyse the bacteria. And so in the panel C, there is lysis of only the pseudomonas. And you see that this initially, like rather, yeah, where you still see the structure of the bacteria, it becomes like a smeared out red kind of membrane, and the bacteria is still visible. And here they actually lyse the E. coli, and that now the E. coli make this kind of smeared out green stuff. So in a sense, now this is a structure that contains the contents of Pseudomonas as a membrane and the contents of E. coli as an interior, but the bacteria are not intact anymore. And then what you essentially have is this, of course, is something like a compartmentalized cell extract made from E. coli, like not a cell extract, it's like a pure, a cell lysate. And in principle, this is also should still be capable of doing gene expression. So one thing that they did here, and there's kind of a lot of other stuff in that paper, and I think that many things they just did because it really looks cool, and it in a sense indicates that you can generate something that really or almost looks like a cell using kind of tricks that in this context. So what I think what they did here is they actually first made these kind of structures where they lysed the bacteria, but then they added bacteria again. So there are live bacteria inside of that structure. And what they also add to the whole thing is G-actin. That's the monomer of actin. And you can polymerize actin into actin polymers, filamentous actin, in the presence of ATP. And apparently when they do that, so the bacteria grow, and it turns out that growing bacteria, and I checked the paper that they cite, growing bacteria apparently also lose some ATP. So the bacteria, in a sense, have a metabolism. You only need to feed them sugar, they grow and release a little ATP, and the ATP is used to polymerize the actin into filaments. And over the course of several days, what they see is the emergence of water-filled droplets they call vacuoles. And because of the growth of the actin filaments, they see a deformation of the whole structure. And here they also have a DNA histone protonucleus. And I think that this is probably the remaining DNA from the bacteria that were present in the beginning, and this is stained with DARPy as usual. So in the end, what they generate is a structure that just superficially looks like a cell with a nucleus, cytoskeleton, and so on. And I think that opinions probably differ what that actually means. It's because, yeah, it's not a bottom-up cell in a sense, but it's making it's more like a maybe Frankenstein type of cell. You put in stuff and something happens. But on the other hand, to say something more positive, I think that on the other hand, this really looks like a cell, and it maybe indicates that that the formation of such structures in the end might be quite natural, that this type of organization is just how it will look like. So that, to my knowledge, is one of the kind of most like recent major achievements in this field. But I would say that this is neither a proto-cell nor I don't know what really it is, but yes, question.
The, the drop is lying on some kind of
Very good question. Actually, I don't know. Interesting. Yeah, usually these things are really very viscous and you would not have them lying on a surface, but maybe they are stabilized by the bacteria. I haven't checked it. I don't know.
And the resulting thing that doesn't have a membrane. So that the
No,
bacteria
it's not I'm actually also not sure what it is. I think that what that stuff is, that must be the remainder of the bacterial cell wall of the Pseudomonas. And uh, so, which would be the remainders of that stuff here. And so maybe there is some peptidoglycan and whatever, but I don't know how it is structured. I guess that it will still be permeable. It's not like I think it's probably a macromolecular, like a meshwork that just like goes around the whole thing. Okay, I was too fast today. I'm done with the synthetic cells. Thanks for your attention. Okay.
Sorry.
Yes, I was waiting for your question. I was waiting. Okay.
Thank you. Yeah, very interesting. What I was wondering was, yeah, as you mentioned at the beginning, this is like bottom-up approach, and I feel like from the chemists and physicists, this is like the approach of those fields, right? In biology or in synthetic stuff. But what I was like kind of missing was like systems biology,
Yeah.
the top down approach. What will be like the top down approach of these things? As you mentioned, maybe it's the minimum cells, but
Yeah.
if you want to make synthetic cells from a top down approach, you will have like the inspiration of biology. Will not
Yeah.
be like completely artificial or the novel life, but I kind of feel like that can help, maybe.
There is a there are top down, of course, approaches, and I would also have another lecture on that, but I was not sure whether I'm going to talk about this or the coacervates next time, or maybe I'm going to make a mixture. The problem is that there's not a lot of work in the top-down approach. There was that famous kind of work by Craig Venter and his group, and and for this specific system, the J. Craig Venter, this Syn cell 3.0, there's actually a computational model for the whole cell. So, and that is more like a systems biology approach then. And I think that people have done that. And I'm also, I think that probably also people try to model, for example, the behavior of one of these bottom-up cells using the typical systems biology tools. I haven't talked about that, but I agree that in principle, it would be better not just to write down a list of components, but then try to simulate how the whole thing, whether it could do what you think. So yes, maybe I don't I'm not up to date in terms of the literature in this field, but yes, and you could also imagine that, of course, with improved computational methods, it is actually tractable to maybe calculate such things at one point. In many cases, one of the problems still is that you don't know all the, like these systems have many parameters and you don't know all the parameters very well. But I'm not sure whether I'm answering your question right now.
Yeah, it's not maybe a question with an answer. But yeah, I was wondering that. Yeah, because I feel like life maybe is more than the sum of the parts. So in this regard, I kind of feel like it's only trying to stick together different parts and hope for the rest, as you mentioned. So
I think that, of course, there are metabolic network models, for example, for E. coli. So they are kind of established, and one could, in principle, use them and erase components or just use the ones that you would need for such a minimal system and simulate that. I think I'm almost sure that somebody must have done that. I just don't have the paper here. Yeah.
yeah, and also about the almost last that the maybe the approach of seeing these things like robots, because I think it's also, as you mentioned, very important because what I think is the per se importance of all of biology maybe is like these self replicating devices like robots, because the chemists, for example, are interested in biology because biology, they have a more efficient approaches of synthesizing things, right? But at some point, maybe we can equiparate to those kind of processes. But the thing that is per se, I think, very important in biology, or the per se importance of these things are that they are self-replicating devices or something like that. But yeah, okay.
Yes.
Okay.
So I could imagine that. Okay, I don't know where the future will lead us, but let's say that, of course, there are kind of connections between chemistry and like chemical synthesis and biology in fields like chemical biology or others. And if you think of people who want to expand the genetic code, for example, or want to incorporate unnatural amino acids into their systems, I could imagine that you would actually at one point create living systems that contain many non-biological components, and they might actually further evolve, and it could be something very different in the end. And that would be more like a living robot than in the end. I guess that's not, I don't know. Yeah, it's also not answering your question, I guess, but it
Yeah. I think it's more philosophical or something like that.
okay, then have a
Okay.
nice rest of